I'm beyond thrilled to have our speaker here today. Her name is Jensen. Her bio is I have to read this though, because I've read it a dozen times and I still don't understand it. Katie is a doctoral candidate in bioinformatics and computational biology at UNC Charlotte. She holds both a BS in psychology and an MS in bioinformatics and genomics from Charlotte. Her work is primarily focused on the shape, function, and changes of nucleic acids in the context of behavior and disease, with a special interest in neurodegenerative diseases such as Alzheimer's. For the past 7 years, she's also served at the UNC Charlotte community as an instructor and mentor. That's that side of it, so she's got a heck of a resume. However, what I wanted to say is we met Katie, Jenny and I, and a few other folks who were at Answer's fundraising event that Susan hosted recently. We met Katie at this event, and it was a wonderful event. It was fantastic. Answer raised a lot of funds for their cause. And for those of you who don't know about Answer Scholarship, Answer was started by Susan Anderson, the executive director, back in 2005, 2006, Susan, is that about right? And Susan saw a need to provide working moms with an opportunity to get their college degrees. That's what Answer Scholarship does. They provide money to moms that are working. Katie is a mom with 2 kids, and they give moms a hand up to get them on their way. It fits in so well with our Empowering Girls and Women initiative. And we were happy to partner with Answer a couple of years ago in providing funds for their emergency fund to help their scholars stay in school. They have an incredible success rate with over 90% of the people that they help, the women and girls that they help, graduating from college. And that's where we met Katie. We found out that she was not only an incredible professional in her own field, but she's a graduate of the Answer Scholarship Program. And she is now one of the mentors for Answer. So she's gone full cycle, and we are incredibly fortunate to have Katie Jensen here today. Thank you for being here, Katie. I'm going to turn the floor over to you.
Yes. Katie, I want to let you know I'll do the program or the presentation, but my computer is kind of acting up, so hopefully we won't have any issues. We will try and see what happens.
We will go with it. It's all good. Thank you.
You can go ahead and share it whenever you're ready, Jenny.
Okay.
Perfect.
You see it?
Yes.
Thank you.
Thank you. Thank you. So thank you all for this opportunity. So honored. So Leave It Better Than You Found It is actually a logo that I have on one of my shirts. I think it's a pretty great way to live life. And several of the points I'm going to touch on kind of are relevant in this context. So, thinking about changing personal behaviors, breaking generational cycles, contributing to important fields in the area of science, giving back, paying forward, all of that. So I'm basically going to give you a brief overview of where I come from, some of the highlights of my story that got me to where I am today, and then I will explain what bioinformatics is.
Thank you.
Yes. Bioinfo, what is the answer I usually get, or the response. And then I will talk a little bit about my research, and I will try to fit it into like 20, 25 minutes, we'll see. All right. How should
Say
we
next when you want me to change.
Okay, got it, next. So I'll go ahead and get started and then click again and then hover over that one. So my childhood to high school was pretty good. I was born in Illinois. My parents were young when they got married, young when they had children, ready to be independent of their own families. So they started off, we moved around a lot. When I was in 2nd grade, we actually moved to Kentucky. I don't have one place that I can say is my childhood home, but if I were to choose, it would be here. This is 120 acres of land outside of a small town in Kentucky. That is where we lived when I was in middle school until I graduated high school. As was tradition for our family for previous generations, my father was a primary earner. My mother stayed at home, took care of the kids, took care of the house, so on and so forth. She did do some odds and ends jobs like babysitting and stuff she could do from home. She did actually pursue higher education when my brother and I were in high school, but did not receive the support that a mother who's raising two rambunctious teenagers should be getting. So she did not finish her program, but she did land a really good job, got promoted, and that is actually what moved us, our entire family, eventually to the Carolinas. Even though all of our needs were met, our basic needs, we didn't always have a lot of extra. But one of the accommodations my parents tried to make was for piano lessons next, or like, for me, I started playing when I was 5 years old. It's a picture of me playing piano. We'll just go from there. It's not that important. So my parents put a lot of, focus on that, making sure that I could take lessons. By the time I was in high school, I was involved with a lot of things. I ran track, played soccer, had a job as soon as I was old enough to have a job. I was in high school choir, competed in junior miss. I did a lot of things. So it all kind of looked good on the outside. But I had also adopted some maybe unhealthy ways of trying to fit in. Those eventually turned into unhealthy coping mechanisms and or escape mechanisms, right? So because of that, it was almost a miracle that I actually graduated from high school with a comprehensive diploma on time, on track. In addition, you can go to the next slide if you want, Jenny. Is she frozen? Okay, and then next. Wow. Junior miss. And one more, and then the next slide. Graduation with my family. Okay. So I was accepted into a school in my mother's hometown in Decatur, Illinois. That's her childhood home, actually. You can do the next two pictures if you want, Jenny. So Millikan University is a private university. They have a great fine arts program. I was accepted into the piano performance and was offered a full scholarship because obviously our family could not afford whatever it was, 30 grand a year for me to go to school. So this is a really big deal as a first generation student to be accepted into this program. But what I will refer to from now on as the monkey on my back was in full force at that point. So I lasted about a year. Dropped out of college, started working. Working 2 to 3 jobs at a time to try to make ends meet. I eventually moved to North Carolina, to Charlotte, to be with my family so they can help me out. And then I should probably do the slides on here too so I can remember the flow of things. At the age of 19, I was a retail supervisor. At the age of 21, I was in restaurant management. Sounds great, right? The success was very short-lived. The monkey on my back, right? Made sure of that. So I hit a pretty low point, and then I found out that I was expecting. If you can go to the next slide, please. Here we go. So this is my firstborn, Kerrigan. I married Kerrigan's father. That was also short-lived. Got to the point where I feared for my life, feared for the well-being of my child, moved back in with my parents, continued working. But that year was a really tough one for me, and I hit rock bottom at the end of that year. Had a lot of nasty consequences that I had to deal with. One of them being losing my license. Kind of got things together, got the help that I needed, got everything together. Things were running smoothly, just the two of us in a one-bedroom apartment. And then I realized that I wasn't meeting my full potential, and that was having an impact on my child. She was dreaming about doing things like being a taxi driver, a bus driver, or a bartender, or working in a retail store selling clothes, which is fine, but I want my children to be able to reach their full potential, and I wasn't reaching my full potential. So made the decision, go back to school. Next, I started at CPCC to get my associate's degree. I was in the Honor Society, I was a VP of Scholarship, here with all the other lovely VPs. So I got my associate's degree, decided to go to UNC Charlotte next. Met this guy, married him. We're still together, going on 9 years. Shortly after we got married, well, actually the year that we got married is the year I started at UNC Charlotte next. And then, after my first year at Charlotte, I found out that we were expecting again. Next. So Elizabeth was born. I remember feeling very excited and also feeling very defeated because I didn't know if I could handle continuing my degree and having 2 children to take care of. I went into one of my professor's offices, shut the door, I was in tears, I was like, I don't think I can do this, I'm gonna have to drop out, I don't know what to do. She was so kind, so understanding, also very firm. And she said, absolutely not, you will not drop out, you can do this, a lot of women with children do this. You can, she told me about different options that were available, and one of them was Answer Scholarship. Next. So I applied and was accepted as a scholar with Answer. This is a picture of our little family with our lovely Susan Anderson at the, I believe this is the family room opening at Atkins Library at Charlotte. Next, and then my mentor was actually Dr. Ann Moore, who is the dean of the library at Charlotte. She helped me through so much, right? I'm forever indebted to Answer and the Mentors for Mom program. You know, not only the fact that I had a mentor, but we had workshops, and I've met so many other amazing women who either have already gone through going to school with children or were going through the process as well. We were all in it together. It was so empowering, in fact, that I decided let's think about graduate school. Next, so I started the early entry master's program in bioinformatics. A year after I graduated with my bachelor's, I graduated with my master's and said, you know what, sky's the limit, let's go to PhD. So that's where I'm at now. Next. And actually about a year ago, I got my license back. I had finished all my classes, really just working on dissertation, and decided it was time to start giving back and paying forward all of this amazing support that I received. So I came back to Answer and I said, can I be a mentor? And they said yes. They welcomed me with open arms. This is my lovely scholar this year, July. She's from Columbia. She has 2 children. She's at CPCC, studying to be a medical laboratory technician, and she will finish this May.
Wow.
Yes, it's truly amazing. And thank you all so much for the support that you've given us. It's such a huge difference and far-reaching, so greatly appreciated. I'm gonna kind of go off of my personal story and start leading into research, introduce bioinformatics. I don't know, where am I at on time?
You get.
Okay, you tell me what I got about 5 minutes. All right, next. So one of the reasons I'm interested in Alzheimer's is because it hits very close to home. So first is my great-grandfather. He was a welder at Caterpillar before he retired. He actually developed this automated process for the welding. It was so helpful that they sent him internationally to other plants to be able to teach this process. He was intelligent, he was well-spoken, he was always well put together. When he was in his 80s, he went to the doctor for a health checkup. Doctor said, you're gonna live to 100, you're in such good shape. 2 years later, he was diagnosed with late onset Alzheimer's. 2 years after that, he was gone. I was actually there, our family was called in, they said, hey, you know, it's kind of near the end. I was still a child, and I watched what happens at the end of this disease. And I will never, well, I may forget it at some point, but as long as I can remember my past, I don't forget that. So that was my first encounter. And then next is his daughter, my grandmother, my mother's mother. I was named after her. Most amazing, sweet woman you'll ever meet, just so thoughtful, took care of everyone around her. Was kind of my second mom for a while. My mother had a difficult pregnancy with my brother. She was so proud of me too when I got accepted into college. That was a really huge moment. And that's actually us in my dorm room. So she was always kind of forgetful, like, you know, and I do this too, glasses on the head, and like, where are my glasses? Or, you know, keys in my hand, where are my keys? It was always super cute, but after my grandfather passed away, it really started going downhill, and we started noticing things that had never happened before. So my family finally moved her down here to the Carolinas and helped care for her, and we were with her until until the end. And then, most recently, my father's mother, my grandmother, has been diagnosed with dementia as well. She's still around, but she kind of has this set agenda in her mind, and her mind loops through it. And she can't access new information, and she can't really think about other information other than what's in that loop. So it's pretty devastating. So this hits close to home, first of all. Next. And also, as you all know, it's very prevalent, especially in America. We've got more than 6 million Americans, give or take, affected by Alzheimer's. So we have this umbrella of dementia, right? And then Alzheimer's falls under that umbrella, and it takes up about 50% of it. And a lot of work, so as we get older, right, we're either going to get cancer or we're going to get Alzheimer's. It's just kind of, not the given, but it's just something's going to happen, and if it's not cancer, the potential of Alzheimer's does increase as age goes on. So that's very scary. And we know that the greatest risk factor is advanced age. We know that it's a sporadic disease, so you can kind of click through the rest of those if you want, Jenny. So there are a set number of mutations that if you get them, you pretty much have Alzheimer's, but that's like a less than 5%. And then as of right now, there are a few therapeutic options, which is where the research comes in. We need more work so that we can do it. From what I understand, you all also support the CART fund.
Yes.
So research is not a cost-free enterprise at all. And we definitely, there's a lot of work that's been done in cancer, and there's a lot of hope there. We're still trying to catch up with Alzheimer's and dementia, and so this is definitely a hot spot, right? So when we think about some of the causes of Alzheimer's, and you all probably know some of this information already, we've got the magic 3 mutations. At the bottom of the graph, it says how many people have the gene. That's a little misleading. We all have these genes. We all have the same genes, but what version of those genes is what differs, right? So it's the variation of the mutation that can make a difference. So if we have set mutations in these 3 genes, it's pretty much a given Alzheimer's is going to happen. The rest of the mutations and variations, yes, can increase the risk, but it also kind of gets to the point where it's a balance between nature and nurture. But we don't know what that whole story is yet, and so that's where a lot of research is being done. So on the nature side, where we're looking at the genomic and what's going on in the body, and then looking at the nurture as well. But it's the environment, what are the lifestyle factors, so on and so forth. And they kind of play off of each other. All right, you can go next. Just gonna do a really quick crash course on nucleic acids in the genome. I think sometimes up in the ivory tower, we forget like how much information is just available to everyone. And even myself, sometimes I have trouble thinking through like all of these processes that are involved. So nucleic acids, we have DNA, we have RNA. DNA is like the storage, it's like a file cabinet, right? So we have, let's say you have a corporation and all of your instructions for all the jobs that need to be done in the corporation are in that file cabinet. That's our DNA stored. The instructions for the body to do what it does and be what it is are in that storage space. But we create new cells. We have organs such as the liver or skin that are constantly replicating or replenishing themselves. Old cells die, new cells are made, so the new cells need copies of that DNA. They aren't going to be functioning, right, if they don't have that DNA. So we have the replication process. Then we also have what's called the transcription process, where let's say you need to make copies of the instructions, and then you also need to make some copies that are going to be sent off to the people in the corporation who need to carry out those instructions to do the jobs that need to be done. That's the transcription process where DNA is transcribed into RNA. And then RNA generally, in this context, is a single, not the double helix like we see with DNA. And that is then turned into, translated into a protein. Proteins are what do all the work in the cell. So they all have set jobs, but they can do their jobs slightly differently depending on what that message is in the RNA. And so what I'm interested in is, well, primarily interested in two things. One is structure. So DNA, when it is separated, the double helix is separated and we have single strands, has the ability to fold into these really intricate structures. And sometimes those structures can be very stable, which could be a good thing because they signal other proteins to do jobs, or it could be not a good thing. Could be detrimental because if the structure is in a place that it shouldn't be, the instructions can no longer be read. And so then things don't happen. Right, so I'm interested in the structure. I'm also interested in the mutations and the variations that occur when DNA is either being copied and little mistakes are made, because that happens. Just imagine taking a booklet of instructions and writing by hand time after time after time, probably going to be some mistakes, right? And that's what happens in our cells. Over time, tiny little mistakes are made during the copying process. Tiny little mistakes can also be made during the transcription process. And then there are, well, I'll talk about the proteins in a minute that change the RNA. But anyway, that is nucleic acids in a nutshell. We have specific locations along the genome that code for particular jobs, and is a sequence of A's, T's, C's, and G's, right? So we can go to the next slide. I will tell you what bioinformatics is. So biochemistry, next, transcription, replication, and then the jobs that are being done. Okay, so bioinformatics is an interdisciplinary field. We have a lot of different disciplines that go in: computer science, biochemistry, biology, so on and so forth. We ask a biological question, we use a biotechnology platform or more than one. Then we have to use computational analysis. So the data that we pull out of, we take our sample, we get the biological components out of it, then we run it through some sort of process or multiple processes, and then those platforms will give us data in the form of files, but it's a lot of data. So a laptop today has what, about 1 terabyte of storage space? Think about one laptop times hundreds, all full of data, okay? Just that we derived from an organism. So we clearly can't do that by hand. We have to write code. We have to use computational resources to be able to analyze the data. And so that's what I do. I write code to be able to analyze all this big data so that we can actually make sense of it and come up with some conclusions. You can go to the next slide. So Dr. Jennifer Weller is my advisor. She's helping me with my dissertation. She is at Charlotte, but she also has become a National Science Foundation Program Director. Brilliant woman, absolutely. Also had a kid while she was going through school. So I totally lucked out in that department, right? Advisor department. A couple of things I'm working on with her that I'm not going to go into detail, but if you go to the homepage of Bioinformatics Department, she and I put a little video together talking about DNA structure and kind of what we do to analyze it. If you're interested in checking it out. And then also we've collaborated with AppState to work on the App Maze project, which has to do with honeybees. And that's a really cool project as well, so you can check that out. What I'm going to focus the most on for the next few minutes is the work that I'm doing in the Funk Lab. So I'm collaborating with Dr. Kristen Funk in biology in Charlotte. And what Dr. Funk's lab is interested in is the three-way interaction between neuroinflammation, which can happen whenever there is, let's say, a viral infection or a lot of stress or anything along those lines. So processes in the brain, the neurons can become inflamed. Maybe the immune response is not what it could be or should be. How is that related to aging? And then how are those two related to neurodegeneration? So this first part of here, the structural and genetic alterations, that's where I come in. That's what I'm looking at in this collaboration. You can do next until you get to the next slide. The Funk Lab put out a few papers in the past few years if you're interested in how viral infection can increase the risk of Alzheimer's. There's a lot of work being done in that area. It's very interesting. So what I'm doing is I'm looking at RNA editing in Alzheimer's. RNA editing is a process that occurs after DNA has been transcribed to RNA that has not yet been translated to a protein. And there are these different proteins that come in and do in-place changes to the nucleotides in the RNA sequence, which will then change the job of the RNA or change the instructions for the protein. Sometimes this is helpful with the ADAR proteins, for example, which are highly expressed in the brain. If there is a viral infection, especially if it's an RNA virus, there are certain sequences that can be recognized, and when the proteins act on that RNA, it basically is trying to inactivate the effects of the virus because it recognizes, oh, hey, this doesn't belong here, let's get rid of it. But it can also act, these proteins can also act on the host genome, sometimes in a positive way, in a helpful way. Sometimes it can be detrimental if there are some issues. So I'm taking data, I'm using data from the ROSMAP project, which basically is this longitudinal study where nuns, priests, monks have agreed to be studied over time. And they get baseline measures, and then over time, biological measures, measures of cognition, so on and so forth are taken. And then at the end, they have donated their organs, especially their brains, for the research. And then that information is pulled out of the brain. So one of the challenges of working on biological brain data from humans is that our neurons don't replenish like the liver or the skin does. So we can't just go in and get a stick to the brain, right, to figure out what's going on. So these are all post-mortem brains, challenge in itself. But I'm looking at the patients that have Alzheimer's but no other known diagnosis compared to the ones that had no cognitive impairment at the end. So we have a control group, right? And then the sequencing data, which is that biological data that we pulled out, use our biotechnology platforms to get the information, and then are storing it in files. And next, just a quick overview of what sequencing is. After we get that data, well, basically, we chop up the sequence into little pieces, and then we take our reference genome, find out where those pieces are located on the genome, and then look for differences. Okay, thank you. My job is to, first of all, find locations where these nucleotides do not match the reference genome. There's a lot of more that goes into that, but that's the essence of it. What type of information or job are these locations responsible for? Some of the work that I've already done, the preliminary data, does suggest that a lot of these locations are responsible for immune response related jobs, which is right in line with the other work that's being done. And then I'd like to go further and find out if there is a change in the structure of the nucleic acids, and if so, is it related to these editing events? So how is that all working together? Next slide. And then finally, after this project is done, which should be this semester, fingers crossed, because my dissertation should be done soon. I'm also interested in, how does the presence of these stable structures and nucleic acids affect the data that we actually receive from our biotechnology platforms? Because if it's in a stable structure and the platform can't read that sequence, well, then we might think there's a mutation there when actually it just couldn't read it. And so this is ongoing work in the Weller lab. We've tested several technologies already, and I'm working on the last two, like the newer technologies. And yeah, it's kind of a big project, but it's been a lot of fun. And then with the honeybee project, in addition to, well, actually, no, this is the honeybee project. So we are wanting to know how variations in the genome are going to be related to the bee phenotypes. Bees actually can be used as a behavior model. So this will be interesting and can be maybe eventually translated back to Alzheimer's as well. But behavior, striking patterns, threat response, resilience, and so on. And then also how structured is the honeybee genome, because we don't know yet. So I kind of threw that in there, like, hey, let's add this to the plate. And then finally, next, and then I've got two pictures. Question, how does exposure to artificial light affect the transcription and translation process of certain genes in fireflies? This is actually the research program of my oldest child.
Oh,
Who is applying at SSM, so the North Carolina School of Science and Math. Wanted to have a research project to be able to talk about. And so the top picture are my two children outside in our backyard with the fireflies we captured. And the bottom is Kerrigan working in the lab on the biological samples that we got from the fireflies. And so this has been a lot of fun for me to be able to start taking what I'm doing and giving my kids the opportunity as well. So,
that is so cool. Wow.
and that is actually it. So
Wow.
thank you all for listening.
So, I have two comments. One comment, I forgot to mention that Rebecca actually worked on Alzheimer's research at Emory University for 10 years before moving here.
Okay,
So
she's
there's
probably the
that.
expert. Not me.
Yeah. But I was going to ask what your kids are doing. So that you answered that already.
Yes.
But I'm fascinated by your group of monks and nuns. And are they like in all one geography, or are you looking at them across, or inviting them to live packs and things like that?
Yes, so I believe they are from different locations. They're different locations, and it was an initiative that was started here. I don't even remember what year it was. It was like early 2000s. I
Any
think.
idea how many, what the size of the group is? That
Thousands at this point. Wow. Yeah, and it's growing. So
that is fascinating.
yeah, it's very cool.
Why did they pick like monks and nuns and priests? Like, why did they go after that group?
That's a great question. I actually don't know for sure what the answer is, but I would assume a couple of reasons maybe. When you do research projects that are involving humans, a lot of times they fall off the face of the planet, they move, they stop corresponding, and so you start losing samples. Well, if you have someone who's committed their life to be in a particular place and to, serve in a particular way, they're going to be there. And so there's potentially that. There's also potentially the assumption maybe that lifestyles are healthier and relatively similar across the board. So you don't have to worry as much as that variation in lifestyle and environment. Unless you think about like geographical environment, which will differ.
That's what I understand.
Yeah.
It's so fascinating. You mentioned post-mortem, that you have to do your analysis of these things post-mortem. Is there a time frame that's ideal for collecting that data, or can it be just forever?
Not really be forever. I don't know what the ideal timeline would be, because I actually haven't done much work with brains themselves.
Sure.
I just know what I've read and what I've heard. I do hope that my postdoc will allow me to have an opportunity to work with brains and such. But I do know they need to be frozen as soon as possible if samples are not immediately removed. Then also, DNA is relatively stable, but it still has a shelf life. It begins to degrade over time. And RNA is even less stable. So RNA can quickly degrade, even if it's stored in certain chemicals, and even if it's frozen -80. So yeah.
Any other questions?
I gotta ask, did the fireflies survive the research?
They did not. The youngest one actually wanted to know if we were going to return them. We were done with them. We're just like, sure, because she's too little to come in the lab with us. So we took the fireflies and immediately put them on dry ice, and then took them into the lab to keep them frozen until it was time to. Thank you, little fireflies. While we catch up your little bodies.
Thank you for your service.
Thank you for your service, your contribution to science. Yeah.
Well, any questions online?
I think you did an awesome job with this. I've always been interested in DNA, and this cleared up some things for me, but there's so much to learn. How much of the sections have been in general what they know what each thing does in your body? Does that make sense?
As far as like the instructions in the DNA,
Yeah. How much of that has actually been translated? or figured out or?
I think the majority of the locations that actually code for genes that have become proteins that are, coded for proteins, I believe that we have a pretty solid grasp on the majority of it in the genome. But then there are these sections of DNA that used to be called junk DNA that we're finding are definitely not junk. Like there's a purpose for pretty much all of it. I mean, nature is pretty perfect, so I wouldn't be surprised if at some point we're like there's a reason for everything in the genome. But that's kind of where I think we are looking for purpose in certain areas of the genome.
Thank
I don't know
you.
if that answers your question, but yeah.
Yeah, it did. Thank you.
Okay, yeah, of course.
Well, Katie, I can't tell you, Susan, thank you, for what you are doing. I think for me, this is validation of what Answer is capable of doing. I think you are an incredible role model. I'm grateful for the changes, the life changes you've made and the inspiration you're providing your children. That's what it's all about. I admire you so much, so thank you. How about and I think we have, one more thing